Thank you for the introduction. I'm very happy to be here today in this symposium to talk about our newly launched product, ChemisTwin, that was developed in collaboration with ACDLabs. And our aim with this product is to revolutionize chemistry by digitalizing analysis control. And I will explain a little bit more about that. We've launched a platform for automatic NMR spectra interpretation with digital reference material. And this is what I am going to give you more insight in today. One thing I also have to remind the audience prior starting, the life science business of Merck KGaA, Darmstadt, Germany, operate as MilliporeSigma in the US and Canada. I am based in Europe and I refer to the company as Merck, as given that I'm based in Europe. So when we started this project, we wanted to imagine a world where our scientists and the scientists that our organization serve would have more time to focus on the analysis result and less on the controls. And by that, we meant finding the correct control for their analysis, purchasing it, maybe waiting for it to be sent around the world and receiving it. And as one of the major reference material manufacturer in the world, we thought we could maybe do better than sometimes these long lead times of receiving your control. So with ChemisTwin and digital reference material, our purpose was to shape the future of analytical testing, making research and quality control in the lab more efficient and more sustainable. So the idea of digitalizing your control is that they would be immediately available. So it would make your work more efficient, and also it would be more sustainable as it would require less chemical from the production of physical reference material to their use, disposal, and their distribution worldwide. And I will explain how we came to this digital reference material concept and how we are now putting it to practice. But first, we had to select an analytical technique to start somewhere, this concept and this journey. And we decided to go for NMR. Why did we choose NMR technique to start with? It's one of the most efficient and widely used technique for structure elucidation and characterization. So when we wanted to develop a tool for automated structure verification, it seems to be a very good technique to start with. What we also thought in terms of the technique itself is that this was a developing technique, a technique of the future, and therefore interesting for this kind of breakthrough innovation solution that we wanted to launch. Also from a technical aspect, the NMR signal intensity are independent on of the chemical structure. They are only dependent on the instrument itself, but not of the chemical structure, which make it comparable one sample to the next on that same machine. So when you think about automation, digitalization, this kind of possible comparison was something we know technically would simplify our life. Similarly, it's known that you can compare, manually we are doing this, you are comparing spectra between the different instruments. So if an NMR spectroscopist is able to compare spectra from a literature with their own spectra obtained on different machine, there's probably some things here that we can automate. The chemical shift of the peaks in an NMR spectra are relatively the same. So again, comparable. And it's not the chemical shift is a measure of a local environment of the nucleus. It's not highly affected by the magnetic field or probe type. So again, from a technical perspective, when we try to have automation in spectra interpretation, NMR seemed like a very good foot in the door for the digital reference material concept, and that's why we selected it as well. After that, we had to assemble a team to work on that novel concept, and this has been a quite worldwide mismatched team. There's been a lot of heads around the ChemisTwin project to give it to bring it to life. Also for the story, this idea came at Merck based on an ideation campaign. So it was really coming from one of our employees having this crazy idea of digitalizing reference material, of sending data instead of sending powder and liquid to our customers. And the idea just grew widely from there. The team members are based in Germany, Switzerland, France, Norway, Canada, India, UK, bringing all these great minds, different culture and different way of thinking together into one solution. There's 8 people on the core team driving every day the project. 11 scientists in total for NMR and IR are contributing to the data collection and to the scientific soundness of it. 20 software developers. And with the partnership with ACDLab, we had 4 scientists from ACDLab contributing into shaping the solution, and they are constantly today contributing to improving it. And 3 development and business service specialists that are also contributing to this project. So quite a wide team that actually has been and is working on developing ChemisTwin. After putting the team together, we decided, okay, we've selected the technology. We now need to select a scope where we think this would be most useful to NMR scientists. And we interviewed over 75 internal, external NMR scientists, try to understand their major pain point that were not solved today with existing solutions. And what we, in the end, decided was that ChemisTwin would be primarily designed for qualitative features, so verifying the identity of a compound, therefore more on this quality control routine testing idea. In terms of type of molecules, we also had to decide to start somewhere, and we start in our really where we have a strong foothold and a lot of reference material available. So pharma testing and other type related to pharma testing activities. And also we are going to offer ChemisTwin and aim it for university, in particular university that are offering NMR as a service. What we are not aiming to do at all with ChemisTwin, and it's not appropriate, is if you're more in a discovery lab or R&D looking at structure elucidation, complete novel compound for which no physical reference or no literature reference exists today. We are not into that direction. I think the NMR Workbook suite is much, much more suited in this case. And ChemisTwin had really no solution in that area to offer. And the automated quantitative analysis is something that is in development and will be released next year, but not suitable as of today. So what is your aim with digital reference material? Imagine if you could confirm a structure with your NMR, so proton NMR measurement, with just a few mouse clicks. No need for ordering a physical reference material, no need for going and check the literature, no need for manually anoding spectra or manually assigning peaks. This will all be done by the software for you. And this is what we want to offer. This is our vision. ChemisTwin is defined as the online platform for automated analytical NMR spectra interpretation. And digital reference material, we want to define it as simply data you can trust. A digital reference material, they are all produced and quality controlled by Merck. They are traceable to their equivalent well-characterized physical reference material. And when available, we are using our certified reference material that are certified under ISO 17034. And we are in full control of the production of this data, the storage, and ensuring that the data is remaining safely stored and not getting corrupted. So we applied our knowledge as reference material manufacturer to the concept of digital reference material. Now let me explain in more detail how we produce those digital reference material and how it's used. And after that, I'll give you a demo directly of the DRM portal, so ChemisTwin, so you can see what it would look like to use it. But here you can see what is in his stomach, as to say. Starting point at Merck, we have a wide library of physical reference material, over 25,000 physical products that we offer in the area of reference material. From this library, we are taking experimental NMR spectra in our own facility. These are mostly 600 megahertz spectra. Then we take this known structure together with our experimental spectra, and we input them into NMR Workbook Suite. NMR Workbook Suite will predict the spectra based on the molecular structure, and then we will correct that prediction manually and optimize it thanks to our NMR scientist here using literature when needed, using 2D data when needed, to really exactly assign and correctly assign each peak of that experimental spectra to match the molecular structure and to teach the software and the algorithm how to read that molecule. Once we have a perfect match between the prediction and the reality, then we do a quality control, a quality assurance release of the data. So it gets verified by an additional 2 people. And once it's released, we are calling this a digital reference material. Finally, when you as a customer go onto ChemisTwin, this is what you see on the right-hand side. You will upload your own spectra. You will select your digital reference material. And here a customized version of NMR Workbook will do the comparison between your sample and the digital reference material data. In this case, ACDLab will only need to predict the difference that can be accounted for your experimental setting, if you have a different solvent and if you have a different magnetic field. But it will no longer do a prediction on the molecular structure. This has already been corrected by the exact experimental spectra. If you have a good fit between the two, then you get a positive interpretation and a positive match of your sample with the selected DRM. If not, then you receive an answer that this might not be a good match or is even a complete zero match. And feel free to also write questions in the chat if you have them. We'll take them at the end. So how will it work now with ChemisTwin in your workflow? How will you use DRM? You collect your sample as usual. So imagine I'm in the lab and I've been given a sample coming from the production line, and I'm asked to confirm the identity of this impurity. I will schedule my NMR test, prepare my sample, and in this case, I would only prepare the NMR sample. No need for any reference anymore. I'll run the NMR analysis, and ChemisTwin takes it from there. And that's what I want to show you now. There is no need for you to open your spectra. You don't need to do any baseline correction or anything like that. You upload the raw data directly into ChemisTwin. So let's look at what this would look like directly into ChemisTwin. I've already logged in. This is a 2-factor authentication login to ensure that you are the right person logging in and that no one else can access your data. I will start a new analysis and select the raw data that I need to upload. In this case, propofol impurity K is what I've been asked to verify. Let's imagine. The measurement I'm uploading right now is a real measurement that we did in our Switzerland facility. Here I can see my experimental spectra. This is mainly for you to check that you've imported the correct file and that the import is looking okay. Like I said before, I am doing here a qualitative analysis. I'm checking the identity. You can see that the raw data file has been read by ChemisTwin and that we've taken out certain part of the data. The title, the measurement ID that you cannot change is the unique link between the ChemisTwin analysis and your raw data, the measurement date, and the solvent. You also have on the right-hand side some non-mandatory field that you can enter yourself. Some of our customers requested for these fields. They are searchable later to find your analysis, so you can enter whatever you like here. Then you will be asked to add a reference. You can add up to 5 references. You can either search for your reference from the list of all the DRMs, Or you can also load to favorite if this is something you're doing often. Let's say I'm doing very often this verification, so I already saved propofol impurity as my favorite. I can also have a propofol and related favorite, for example, where I'm saving several compounds here that I want to check at once. But for the sake of time, let's do it with one DRM today. If you look at the detail of a digital reference material, you will see that they have a unique identifier. So this is the DRM product number. You can see the CAS number, and you will always be able to see from which physical part number this measurement was done and the quality grade of that part number. So this is a certified reference material according to ISO 17034. And what you can see at the bottom left is the experimental spectra that we obtain from that measurement. So this is our own internal data. You can also see information about how we acquired this data, information about our instrument. So in this case, it's a 600 megahertz, that's what we said, and some information about our methodology to acquire it. So I'll say that this is indeed the reference material I was looking for, so I will run the analysis. And now you may notice here, powered by ACDLab, this is when our customized version of NMR Workbook is working in the background, checking the correspondence between the sample I've just uploaded and the digital reference material. It can happen that the analysis takes a little bit longer than expected. It's usually between 10-20 seconds, but can go up to a couple of minutes. But you don't have to stay on the screen. You can also exit, go back to your dashboard, start the next analysis that you need to do. So you can upload them like this in batch, or go and review some already uploaded analysis. And you see, as I exited, I already got here a notification that my analysis is completed. So at the top, you see the title, propofol impurity K, but what interests me really is to see the results. So here, I can see that my sample was verified against the DRM for propofol impurity K. Don't ask me to read that other name here. And was given a match factor of 100%. With ChemisTwin, we consider that match factor above 92% falls under satisfactory result. We did do a statistical measurement to identify where that match factor limit should be from this 3-star recommendation. And that's the confidence intervals that we are giving. We would consider a questionable result between 92 and 75% and unsatisfactory anything below 74. For questionable, we would ask you to revise the data below that I'm going to show to then make the call, is this a closely related substance or is it truly the substance that I want to see? And here you will have a comparison of the reference spectra that is in pink and your spectra that is here in black. You can see them one above the other like this, or you can see them as overlay as well. You will notice that in your spectra, there are still the solvent peaks that have disappeared from the predicted reference. You can zoom in to really compare and check if you need to. You'll have the chemical structure here of the DRM, and then details on the table of assignment comparing your sample here with the digital reference material on the right. And when there is differences, they are highlighted in yellow here. So for this analysis, it's pretty straightforward for me. I am satisfied with it, and then I will approve it. By approve it, you are electronically signing the report of analysis. In this case, I choose a 2-step approval. So this would normally go to one of my colleagues or my manager for a second review and a second approval, because a lot of our customers need to have 4I principle on all of their analysis. You can disable this if you want, but in my case, I'm also an admin, so I have the superpower to approve myself. And once an analysis has been fully approved according to the SOP, then it will state your compound was verified. This is considered as complete. It can no longer be modified or commented on, and will be saved in ChemisTwin. And you can create the report of analysis. It will contain exactly the same information that I showed before, plus information about who confirmed and reviewed the analysis with your unique ID number in ChemisTwin. So this is what we consider equivalent to the electronic signature here. This report can be shared via email, or you can download it as a PDF to save it to wherever you need it to be saved and shared with. On my dashboard, you can see below, now the propofol impurity appears in my completed analysis, and you can at any point in time go back and check your report again. So this was the look into ChemisTwin, and this was the demo. Let me just give you a couple of summary why to choose ChemisTwin and what it would be interesting for you. We have, and we are aiming to have, the most common small molecule used in pharma application as digital reference material at the moment. So the library size is growing daily. So at the time of writing the presentation, it was 800. Today, it's 1,300 compounds that are existing as digital reference material. We've also released a theoretical spectra with lower quality into the platform, so you can continue to use it even if there is no DRM available. But I'll let you discover that if you try it. In terms of precision and trust, digital reference material are undergoing QC inspection and QA release. So in total, it's actually following a 6I principle. One analyst is doing the assignment in NMR Workbook and describing the DRM, a QC inspector and a QA inspector are then reviewing the data. So it makes 6 eyes on each digital reference material prior to them coming into the database. We also consider that using a matching algorithms as ACDLabs NMR Workbook will provide more standardized and consistent result than manually comparing spectra. DRMs are according to ISO 9001 as well. And in most cases, you will have result in less than 1 minute. I think it was the case today, or close to 1 minute. Your database will be always up to date. ChemisTwin is a cloud solution. And like I said, the database is growing daily. And as soon as a DRM enters the database, it is then offered to all ChemisTwin users. You will save time when compared to manual structure assignment or when compared to using a physical reference material. For the manual structure assignment, we compared using ChemisTwin versus our normal way of working in one of our facilities in the US. And they reckon that on average, it could save them, it would go 3 times faster using ChemisTwin versus their normal process of looking at reference spectra and manually comparing them. It is also more sustainable when compared to using physical reference material, and we are studying this, so I can release an accurate number there soon, but not quite yet. And then I hope that you've seen that through the demo, the software itself is really convenient to use. There is no complex training needed, nothing like that. Finally, what I want to say, you can register for a free trial anytime. You have 45 comparisons for free. You only have to go on the URL provided below on sigmaaldrich.com/ChemisTwin. We have to gather some information from you, your lab and your email address, and from that we can create you a free trial account so you can go and experience it by yourself. And I would like to thank you for your attention and see if there is any question that I can answer today, any topics that we would like us to discuss.